Much of what we learn in biochemistry for one species applies very well to others. Biochemical reactions, metabolic pathways, and processes have a great deal in common across all living cells. DNA replication in the cells of plants and animals is, in terms of the overall mechanism, quite similar to bacterial DNA replication. But DNA replication in plants and animals merits separate consideration. Plant and animal cells are more complex than bacteria, and there are distinctive and significant features of their DNAs that pose challenges to DNA replication. Of course, cell division is the reason that DNA has to be replicated in the first place. But what has to happen for cell division in unicellular organisms like bacteria is significantly different than in multicellular eukaryotes like our cells. Bacteria divide simply to make more bacteria, and unless there's a lack of nutrients, cell division can go full speed ahead all the time. However, in multicellular organisms, uncontrolled proliferation of cells would be detrimental. So for plants and animals, cell division must be tightly regulated. In this lecture, we will consider the cell cycle of eukaryotic cells like our own and the role of the cell cycle in DNA replication. We will discuss the proteins involved and the challenges of packaging of eukaryotic DNA with proteins into linear chromosomes. We'll also see how a quirk in the replication of linear DNA leads to shrinking of chromosomes over time. It also turns out an enzyme that was hoped might keep us forever young is instead working against us in cancer cells. And we'll learn how researchers hope to use knowledge about chromosome shrinkage from their ends to help children with progeria, a disease of premature aging, and to develop therapies for cancer, heart disease, and Duchenne's muscular dystrophy. In eukaryotic cells, replication or synthesis of new DNA is intimately linked to the cell cycle, which serves to prepare the cell for division. The cell cycle is a series of stages that is stringently regulated to ensure that cell division occurs only when appropriate. We represent the cell cycle as a circle. The M phase of this circle indicates that a cell has undergone a brief period of cell division, also known as mitosis, followed by a long I phase called interphase. Interphase is divided into 3 stages during which cells take in nutrients and grow and replicate their DNA, and then prepare for cell division. During the first phase, called growth phase 1 or G1, proteins are synthesized and cellular components are assembled. Towards the end of the G1 growth phase, cells decide whether to enter an inactive, quiescent phase called G0 or proceed to synthesis in the S phase of DNA replication. Cells that proceed towards S phase must prepare to replicate their DNAs so they can divide. Once in S phase, DNA is replicated, setting the stage for the next phase, G2 or growth 2. In this phase, the replicated DNA is checked to ensure that it has been correctly and completely copied, and the cell makes proteins that will be necessary for cell division. When everything checks out, the cell enters M phase of mitosis, where the cell divides, after which the cycle begins again. The cell has various mechanisms called checkpoints that allow advancing from one phase to the next only after establishing that everything is in order. One checkpoint in late G1 checks whether the cell's DNA is damaged. If there's damage, then the cycle stops and the damage is fixed. Replicating damaged DNA would pass mutations onto daughter cells, and this could contribute to the development of cancer. The progress of a cell through the cell cycle is controlled by proteins called cyclins, which bind to a special protein kinase called cyclin-dependent kinase, or CDK. The kinases are active only when they are complexed with cyclins. As you have learned, kinases bring about many changes within cells by phosphorylating target proteins. Different complexes of cyclin with kinases trigger different steps in the cell cycle. For example, the transition into the S phase is mediated by a CDK enzyme we call SCDK, which activates other enzymes for DNA replication by phosphorylating them. The kinase is dependent on the cyclin, so once the kinase has transitioned the cell into the next stage, destruction of the cyclin inactivates the kinase. Okay, so eukaryotic cells go through an ordered series of phases in preparation for cell division, and the synthesis phase, known as S phase, is the stage at which DNA replication occurs. But replication happens only if DNA passes damage inspection. If damage is detected, a series of events arrest the cell cycle. First, the levels and activity of a protein called p53 increase. p53 in turn dials up the production of a protein called p21. p21 then binds to the S-CDK and inactivates it. This prevents progression into S-phase replication until the damage is addressed by cellular repair systems. If the DNA damage is too extensive to fix, p53 will instead trigger cell death to prevent the proliferation of a cell with badly damaged DNA. However, cells that do not have functional p53 may continue to replicate their compromised DNA and proliferate. It is not surprising that over half of all cancers have mutations inactivating the p53 gene. If the DNA passes inspection at the G1-S checkpoint, cells move into S phase. Here, DNA replication occurs. As in bacterial cells, replication begins at sequences called origins of replication. These, let's recall, are sequences in the DNA bound by specific proteins that pry the two strands of the template apart. One consideration for DNA replication in eukaryotes compared to prokaryotes is the vastly different sizes of the genomes to be replicated. Bacterial chromosomes have a single origin of replication, but this is not sufficient for larger eukaryotic organisms. A human cell must replicate 6 billion base pairs of DNA to divide. If the rate of replication is 1 nucleotide per second, it would take approximately 190 years to complete this task, using only a single origin of replication. But human DNA is divided up into 46 chromosomes, so that helps a little. However, if we had one origin for each of those 46 chromosomes, it would still take 4 years to finish replicating the genome to allow the cell to divide once. So how do our cells get all their DNA copied fast enough? It turns out our DNA has 30 to 50,000 origins of replication, and each of these opens up a replication bubble from which replication is initiated in both directions. And what do the sequences at the origins of replication look like? Well, in some eukaryotes, like yeast, origins of replication are AT-rich, just as in bacteria. But in humans, origins of replication are often GC-rich. GC base pairs, of course, are harder to separate than AT base pairs, having 3 hydrogen bonds between them. But there's another feature of eukaryotic DNA. It is packed with histone proteins into the string of bead structures. For humans, unlike bacteria, the main consideration is not which bonds are weaker, but which bonds are easier to access. Some GC-rich regions tend not to get spooled onto histones and remain positioned between the nucleosome beads. Thus, they are easier for origin recognition proteins to access. The synthesis of DNA occurs during the S phase of the cycle, but it is in the preceding G1 phase that a pre-replication complex is assembled at the many origins of eukaryotic chromosomes. During the G1 phase, complexes of proteins called origin recognition complexes, or ORCs, bind to origins. This is followed by binding of proteins called licensing factors to the ORCs. Licensing factor proteins prevent runaway replication and ensure that each origin of replication only fires once. That is, DNA replication initiates from each origin only once per cycle. Licensing factors are made in the G1 phase and help initiate replication, after which they are immediately destroyed, removed, or inactivated. Thus, once they've helped initiate replication at an origin, they are disposed of to prevent each origin from being used again in that cell cycle. Replication forks in eukaryotic cells are almost identical to those in prokaryotic cells. There are many more origins in eukaryotes, but replication is bidirectional at each replication bubble, just as in bacteria. Eukaryotic replication forks have the same basic scheme of leading and lagging strands as prokaryotes, though the division of labor of DNA polymerases is different. DNA synthesis is always 5' to 3'. This creates the same challenge as bacterial DNA replication, namely that the lagging strands get made in pieces or Okazaki fragments, with new primers laid down as the replication fork opens up. Eukaryotes have many of the same enzyme activities for replicating DNA. They include helicases for unwinding strands, topoisomerases for preventing unwinding of strands from becoming overwound, single-strand binding proteins, a primase, and a DNA ligase. Not surprisingly, eukaryotes also have DNA polymerases, but whereas E. coli and other bacteria have 5, eukaryotes have at least 14 of them. 3 of those are relevant for us here. They're called alpha, delta, and epsilon. They divide labor amongst themselves a bit differently from the polymerases in E. coli. Polymerase alpha is a maverick with its own associated primase that makes an RNA primer. It's also error-prone with no proofreading function. Neither does it use a sliding clamp seatbelt, so it falls off DNA easily. Now this turns out to be useful because polymerase alpha can readily be displaced by the other polymerases for synthesizing new strands. Eukaryotic cells have different polymerases for leading and lagging strands. Polymerase epsilon replicates most of the leading strand, like polymerase 3 of E. coli. Polymerase epsilon has its own seatbelt, a protein called pCNA, to keep the polymerase from falling off the DNA. Meanwhile, synthesis of the lagging strand is largely a function of polymerase delta, which also uses a pCNA seatbelt and also has the ability to proofread and correct its work. Polymerase delta also plays a role in RNA primer removal, a task for which it is helped by other proteins. Okazaki fragments are joined by a DNA ligase just as in bacteria. DNA replication stops when 2 replication forks meet each other or when the end of a chromosome is reached. Now, so far the eukaryotic process is very similar to what we saw in bacteria, though the cell cycle for eukaryotes adds a wrinkle. But there's a more significant difference. Eukaryotic DNA is packaged together with proteins to make nucleosomes, and nucleosomes are tightly compressed to make chromosomes. These packing proteins get in the way of DNA polymerases that replicate DNA or RNA polymerases that make RNA from it. You can think of the resulting nucleosomes as building blocks of chromosomes. Chromosomes contain DNA packed tightly, just like 500 feet of kite string can be packaged in a small container on its own spool. When cells replicate their DNA, it must be unwound from the protein spools to allow access to the polymerases and other replication proteins. And as the original DNA is replicated and 2 new DNAs emerge, each of them must be repackaged into nucleosomes. The nucleosomes ahead of a replication fork are disrupted as the replication machinery moves in and begins to open up the template. 2 displaced histones associate with the clamp, which helps the nucleosomes reassemble on the 2 new DNAs behind the fork. These histones deposited on the DNA are then joined by 2 other histones to complete the formation of the protein spool for the nucleosome. Obviously, to package both DNAs, new histones will also be needed, and these are synthesized in large quantities in the S phase. There does not seem to be a pattern to the distribution of old and new histones on the DNAs. Some new nucleosome cores are part old histones and part new, while others may be composed of all newly synthesized histones. In either case, both DNAs are wrapped up again with the histones forming nucleosomes on each daughter DNA behind the replication fork. And that's a wrap, except for one crucial issue. The chromosomes in eukaryotic cells cannot be replicated all the way to their ends. Unlike the circular bacterial chromosomes, our chromosomes are linear, and this presents a big challenge. To understand what the problem is, we need to go back and look at the replication fork, focusing on the RNA primers. As you know, each Okazaki fragment begins as a short stretch of RNA which must later be removed. You also know that the replication of DNA can only happen from the 5' end to the 3' end. What you see then is that when you get to the very end of a linear chromosome, the RNA primer can be built as usual and eventually removed, but this leaves a short stretch of the template unreplicated. The overhanging end of the template cannot be copied because we've run out of room to place an RNA primer from which DNA polymerase can extend. So the new DNA is a bit shorter than the original template. When the new DNA is copied in the next round of replication, the same thing will happen again. And the DNA made in that round of replication will be even shorter. As a result of this end replication problem, the loss of nucleotides at the ends of each chromosome with each replication cycle leads to physical shortening of the chromosomes. The greater the number of cell divisions, and thus the more times DNA is replicated, the shorter the chromosomes get. So if you compare the chromosomes of a child with that of her grandparents, Grandpa and Grandma's chromosomes will look like someone threw them in the dryer in the hottest setting. So, you may be thinking, if DNA is the information carrier of the cell, and all of it doesn't get copied, aren't we losing genetic information every time our cells replicate their DNA? While this is a concern, over a span of years, our cells have evolved mechanisms to deal with it, though. For starters, our chromosomes have DNA sequences at each end that do not encode proteins. That is, at the beginning and end of each DNA molecule that makes up a chromosome, there's no information that is necessary for making proteins the cell needs. Instead, there are thousands of repeats of a short sequence that is rich in T's and G's. In vertebrates, each repeat has the sequence TTAGGG. These repeated sequences at the end of chromosomes fold over, forming a loop-like structure that is stabilized by the binding of proteins, effectively tucking the ends of chromosomes out of sight when replication is not happening. These structures at the ends of chromosomes are called telomeres, from the Greek telos, which means end, and meros, meaning part. So a telomere is literally an end part. So those repeated sequences at the chromosome ends serve as protection in the sense that chromosomes could lose a few of them in each round of replication without losing important information. You might think of these sequences as blank pages at the start and end of a book. If you lost a blank page at either end, you'd still be able to read the whole story. It's only after you've lost all the blank pages that you start losing something important. And that's what happens in our cells. Our chromosomes shrink with each round of DNA replication, but we're okay till our cells have divided so many times that further shortening of the chromosomes starts to be a problem. This helps explain an observation first reported in the 1960s by Leonard Hayflick, that human cells that are grown in culture cannot divide indefinitely. He was studying somatic cells, that is, cells that are not eggs or sperm. What he noticed was that cultured cells would divide a certain number of times, but then stop dividing and generally begin to show signs of cellular deterioration. The observation became known as the Hayflick limit. Until that time, there had been a guess that maybe individual cells from multicellular organisms could live indefinitely. Instead, not only did they stop dividing, it turned out that when they stop is based on the age of the individual they were obtained from. Somatic cells from a baby could divide 80 or 90 times in culture before stopping, but somatic cells from someone in their 70s would only divide 20 to 25 times before running out of steam. This suggested that the shrinking of our chromosomes is kind of a biological clock, one that keeps track of how many times our cells have replicated their DNA and divided. And once a certain number of rounds of replication had occurred, we'd begin to lose important information, and our cells would stop dividing and eventually die. Now, two questions immediately came to mind. If our chromosomes shrink with age, how do parents pass along full-length chromosomes to their children? If each generation were born with successively shorter and shorter chromosomes, then the human race would have died out. Another very different question is, if our telomere sequences did not shrink, would we live forever? Or maybe at least stay youthful longer? The answer to both those questions lies in a second mechanism our cells use to deal with the end replication problem. This is a job for telomerase, an enzyme which can add telomere repeats to the ends of chromosomes. When telomerase is active, it adds many copies of the repeated sequence to the ends of chromosomes, ensuring that the chromosome shrinkage doesn't affect important information. Telomerase is a fascinating enzyme. It has two components that allow it to add the repeating sequences at the end of chromosomes: an RNA known as telomerase RNA component, or TERC, and a protein called the telomerase reverse transcriptase, or TERT. A reverse transcriptase is a special kind of DNA polymerase that can copy an RNA sequence to make DNA from it. Let's take a look at how these two parts work together to add telomere repeats. Like all DNA polymerases, telomerase adds nucleotides to the 3' end of a strand. Each end of a linear chromosome has one free 3' end, due to the incomplete replication of the lagging strand, as we've already seen. As you see, the TERC sequence of RNA is complementary to the telomere repeats. The very end of the TERC base pairs with the last telomeric repeat sequence on the 3' end of the overhanging strand. So all the rest of the TERC is available to act as a template for the reverse transcriptase to extend the end and make more DNA. Once a complete TERC has been copied, the entire complex slides down and repeats the process. Telomerase adds many repeats before it eventually dissociates from the end of the DNA, having extended it by many nucleotides. As a result, there is now plenty of room to place an RNA primer on the newly extended strand and complete replication of it. So no information is lost, and the chromosomes remain the same length at each round of replication. Now wait a minute, you're thinking. If there is an enzyme that maintains chromosomes at the same length, why do they get shorter? Well, that's because the magic of telomerase is active primarily in our egg and sperm cells, and in stem cells, which are a stock of undifferentiated cells. Our somatic cells, with few exceptions, like some of our immune system cells, make little or no telomerase, even though all of our cells have the genes for it. Because telomerase is active in egg and sperm cells, they retain full-length chromosomes, ensuring that the next generation starts out with lots of the telomeric repeats at the ends of their chromosomes. But all the somatic cells not making telomerase have chromosomes that do shrink with age. So yes, telomere shortening has been linked to aging as well as to lifespan, even though there are also other factors such as oxidative damage that also affect aging. So could we turn the telomerase back on in our cells, recover our lost telomeres, and recover our youthfulness? If all our cells made plenty of telomerase, would we be essentially immortal? Well, not so fast, says Mother Nature. One of the useful things about cells eventually ceasing to divide is that this provides a built-in limit on cell division. In fact, telomerase that stays turned on with no built-in limit on cell division is often found in cancer cells. In rapidly dividing cells, telomeres would normally shorten swiftly to the point where they would have to stop dividing. It is the reactivation of telomerase that allows cancer cells to keep proliferating without anything to check their division. These cells are immortal. Even if they kill their host, they can be kept alive in a lab indefinitely. Non-cancerous cells can't be kept alive like that, as we've seen. So they really are potentially immortal, but they're also cancerous. Always a catch, right? But maybe we can use this information in a different way. Since cancer cells express significant amounts of telomerase, unlike most normal cells, drugs that inhibit telomerase would be useful against cancer cells. And because close to 90% of all cancers express telomerase, a telomerase inhibitor might be useful in treating a variety of different cancers. Indeed, in lab experiments, telomerase inhibitors have been shown to be effective in killing cancer cells, and some are in clinical trials. Telomerase activity is also inhibited by compounds found in foods like curcumin from turmeric and allicin found in garlic and onions. Green and black tea also have a phenolic compound that inhibits telomerase called epigallocatechin gallate. Populations that routinely include these in the diet may have a lower incidence of cancers. But such preventative measures cannot replace cancer therapies for those who already have cancer because they do not provide sufficient inhibition of telomerase activity once cancer is underway. Although the relationship of telomere length to aging is not fully understood, people with longer telomeres seem to live longer. Conversely, telomeres are much shorter than normal in children with a mutation that causes progeria, a disease of premature aging. Now, children with progeria begin developing symptoms like baldness and severe cardiovascular disease as early as age 2. Most do not live through their teens. Shorter telomeres have also been found in boys with a form of muscular degeneration called Duchenne's muscular dystrophy. Even stranger is the finding that stress can lead to shorter telomeres. The length of a baby's telomeres has been found to be related to maternal stress during pregnancy. Children in orphanages in Romania were found to have shorter telomeres than their peers who were placed with families. So is there anything we can do to prevent telomeres from shrinking before their time without creating conditions favorable to cancer? In 2015, scientists at Stanford University's School of Medicine showed that it was possible to temporarily increase the activity of telomerase in cultured cells, leading to telomere extension over a period of about 48 hours. After this time, the cells went back to not making telomerase, but by then the telomeres had been extended significantly. These cells were able to divide more times in culture than comparable cells that did not have their telomeres lengthened. A similar experiment was done using cells from children with progeria. In 2017, scientists from the Houston Methodist Research Institute were able to dial up telomerase activity in these cells. They found that even a temporary increase in telomerase was able to turn back the cellular clock, effectively reversing the signs of aging in the cells. The hope is that these experiments can be stepping stones to therapies for progeria as well as for Duchenne's muscular dystrophy. Interestingly, increasing the expression of telomerase following a heart attack or myocardial infarction was shown to significantly improve heart function and reduce mortality from heart failure in mice. Eukaryotic DNA replication is in many ways a scaled-up version of prokaryotic DNA replication. However, the issues associated with the linear ends of eukaryotic DNAs impact aging and cancer and make this otherwise esoteric topic into one of the most exciting areas of research. Active research in telomerase is opening up new doors for treatments for cancer, the potential for life extension for sufferers of numerous diseases, and it will likely provide insights into the process of aging with the potential to affect everyone. At this time, we don't know if compounds that affect telomerase synthesis and expression, like resveratrol found in grapes, or genistein, a phytoestrogen in soy products, will provide the health benefits some proclaim. What we do know, though, is that there is no harm in things such as physical activity, eating fruits and vegetables, and relaxing, all of which do increase telomere length in humans.